So, if we're going to change this, we're going to have to come up with a better system. Otherwise, it's not going to be adopted. Buckminster Fuller said it best. So biology has shaped almost every facet of this planet's development for the last 4.2 billion years. It has accomplished this by doing two things: increased system biodiversity. After every extinction event on this planet, there's been an increase in biodiversity. Now, if you're part of the group that gets to be in that survivor group, that's a good thing. We need to be concerned about that, because we may not be in that group. The Earth is going to be fine. Also, it's done this by improving the efficiency and capture and storage and utilization of solar energy. This is what the past has looked like for the last 600 million years. You can see there's, on the black line, let's see, does this have a laser? Oops, nope. The black line on this chart is your CO2. The red line is your oxygen. The blue line is the temperature that's been on this planet. As you can see, we've had CO2 up to 7,000 parts per million. We've had temperatures up to 25 degrees C. Oxygen content up to 35% and down close to 10. We are here now. We're about 14 degrees C. Our CO2 is about 400 parts per million. You can see at each extinction event at the bottom, we have a rise in CO2 in the atmosphere. Let's see what biology has done. You'll see decreases all along this. Let's see how biology has affected this. Starting the Cambrian explosion, you see a sharp decrease because we had the development of coral shelled animals, and they were taking calcium carbonate out of the seawater. You have your first land plants, Guamaraceae, right there about 500 million years ago. You have lignin and vascular plants developed during the beginning of the Silurian. Your coral reefs came in at the, excuse me. Yes. At the start of the Permian, we had 8-meter tall fungi, progenitosperms, roots, leaves, and seeds. Now this led to the availability and opportunity of plants to colonize the full planet instead of just being around water bodies. You can see sharp declines. We go into the Carboniferous period. You can see increased carbon sequestration. You can see atmospheric CO2 reduce. Along comes lignin-degrading microbes. You can start to see a change, an increase in CO2 here. Then came in your conifers and geckos, evergreens, and each of these adaptations is an increase in the ability of these systems to capture energy and store it and utilize it. Angiosperms come along, your coral reefs, and finally, about 55 million years ago, we had a cooling period, landmass positioning changes, mammals, ungulates, or your grazers, and grasslands developed. We didn't have grasslands till 55 million years ago. But it was this system that reduced our CO2 down to the 400 or less than 400 parts per million. And it stored that carbon in soils. And some of the soils that we had were over 6 feet deep with high carbon content. So you can see biology has the power to make these changes. The way it's done it is microbes are the backbone of every organism on this planet and ecosystem. You are about 10 trillion cells. You have about 90 trillion microbes that associate with you and help you. You have about 30,000 genes. They have about 8 million genes. You are not alone in this venture. What do they do for you? They digest your food. They generate nutrients for you. They do vitamin synthesis. You can't get these in your diet. They detoxify carcinogens. They prevent allergies, skin diseases, asthma. They comprise 60% of your immune system. They control your appetites and cravings. They turn on and off genes in your body that regulate brain development, anxiety, depression, autism, arthritis, and emotional behavior. Do you feel like you're in control? So what happens when these communities get disturbed? Clostridium difficile is an infection of the intestines when you get an imbalance in the microbial community structure. Also, IBS and Crohn's disease. They found that with simple inoculation or fecal transplants from a healthy donor, people that have suffered for years and decades with these diseases can be cured within 24 hours. They see cure rates for Clostridium difficile over 92%. Also on Crohn's disease, they're seeing a 62% improvement in people's condition. We've kind of done this to our agricultural systems. Everything we've done, the plowing, the biocides, all the things that we've, the single species crops, all of this has led to a disruption of the microbial community in these soils. What if we did something like a fecal transplant? Can we cure them? Let's look at how nature has set this up first. In a bare soil parent material, you see a predominantly bacterial system. As you move up this ladder of succession, you see a shift towards fungal dominance. You also see a shift in the ability of these systems to increase net primary productivity. In that 100% bacterial, your desert soils, about 90 grams per square meter are produced per year. Up to your old-growth forest, your most productive ecosystems of about 2,200 grams. We are here in agriculture. We grow weeds really well. Of course, we don't eat weeds. They have a purpose. They're there to improve that soil. In our region, if you scarify the desert, tumbleweeds will come in and grow. They'll grow for about 3 years, and then they won't grow anymore. They have a purpose in improving that soil to the next trophic level or next species that comes in to improve it step by step up this chain until you reach your conifer forest. What we need to be is a fungal bacterial ratio about 1 to 1 or up to 5 to 1. That's what we're going to do best for the crops that we like to grow. But we need to understand agriculture is controlled disturbance. These are the organisms that did that controlled disturbance for our grasslands. They built up these soils. They built up fertility in these soils. They built up carbon in these soils. And what we need to do is try to mimic these animals in the process. We realized that the way they did it was they would come in, they'd graze only about 25 to 30% of the forage that was there. They were being herded by predators, so that kept them moving. They would leave their dung. The dung beetles, your insect community, would roll that dung up into little balls, stuff it into the ground, and it would compost in place. Well, we need to try to mimic that in our agricultural systems if we don't have the grazers. So, in a project with USDA, I was supposed to be working on how to compost dairy manure and come up with a good product. The problem with dairy manure is it was very saline, and most people that ended up composting it, it ended up more saline, or as saline as what you started. It was about 30 millisiemens conductivity in the dung, or in the dung from the dairy manure, and that would end up with about the same in the end product, the people that had this a decade before me. They were doing it with a windrow composting process, so we tried a static composting process to see how that would reduce the salinity. Well, we were able to reduce the salinity from 30 millisiemens down to less than 3. We were able to use less water doing it, and we were able to do it in a shorter time period. But what we got out of this product was a fungal dominant and biologically diverse compost. This came in very handy when we started doing plant tests to see how effective it was. But what we noticed first through the composting process was a change in the diversity. This is a 16S assay on the bacterial community of the composting process at 1 week. This is 22 weeks. This is when everybody says it's ready to go. So you can see the diversity has increased since that first week. And this is at 1 year. So you are increasing diversity in this system the longer you let it compost and the more you let it mature. So we use this compost to mimic what our soils look like. On the top left, you see a fungal bacterial ratio of 0.04. This is a low carbon soil, bacterial dominant, all the way up to a fungal dominant, 3.68 to 1, and a higher carbon percent. We were looking at how the carbon was partitioned into your root carbon, shoot carbon, fruit carbon, new soil carbon, your respiration carbon, and your total carbon that would go into nitrogen fixation. Now, carbon that goes into nitrogen fixation is very expensive to fix nitrogen. It takes about 12 units of carbon to fix 1 unit of nitrogen. You can see in a bacterial dominant soil, low carbon, only 3% of the carbon that plant captured went in to the plant. As you improve, or as you increase the fungal to bacterial ratio and increase the soil carbon, we saw up to 56% of the carbon go into the plant. That's where we are in agriculture today. We are low carbon and we are bacterial dominant. So you have a choice. Which one would you rather farm on? Where do you want to put your energy? You want to be up in this range, so you're getting more return on the effort you're putting in. So in this process, we see an increase in net primary productivity. To give you a kind of an estimate of how things are in this planet, in the old growth forest or tropical forest, you see about 2,200 grams of dry biomass per square meter produced. In our cultivated land, the land that we fertilize, the land that we plow, the land that we irrigate, you see only about 600 grams of dry biomass per square meter produced. We're seeing, with the application of this compost at about 400 pounds per acre, up to 2,200 grams increase in productivity, with the potential of 3,186 grams of dry biomass per square meter, just by restoring the diversity of the microbes in the soil and changing that microbial structure from a bacterial dominant to a fungal dominant. This is the application rate we started with. It's about 400 pounds per acre. We only applied once at the beginning of this 7-year trial. And what we're doing is we're growing crops, cover crops, green chopping them, pushing them into the soil, and growing another crop. We designed this experiment to see what kind of carbon increase rate can we see in a soil when we dedicate all its energy that it captures back in. After 1 year of the BEAM application, it's a biologically enhanced agricultural management. You can see, compared to our control of a conventional field, that we see a 5 times increase in productivity. At year 7, we're up to 1,141 grams of dry biomass per square meter. So over this period of time, we've increased the productivity of this land 5 times. We did the desert plot just the opposite. We wanted to grow a cover crop, cut it, haul it off, and grow another one. We wanted to tax this system as much as possible. What we want to see is, on both ends, if we put everything back and took everything off, where in the middle is that sweet spot where we can grow the crops we need and then improve the soil at the same time. The first season of this cover crop on the desert soil, we saw about 800 grams of dry biomass per square meter. This is the second season at 1,608 grams. You can see we've captured 486 pounds of nitrogen per acre on this plot at the rate the plant growth is. This is the third season, and each year this thing has improved. We're seeing 2,190 grams of dry biomass per square meter at 583 pounds of nitrogen per acre. This is about any nutrient applications. This is just using biology from the compost that we make. This is what the winter cover crop biomass on the left. You can see the yellow is the control. The green is the treated with the compost. You can see the difference in increase. We started out with about 492 grams of dry biomass per square meter. Thank you. We're ended up over here at 3,186. Now remember, 2,200 grams is the most productive ecosystem on the planet, and yet we can see over 3,000 grams of dry biomass by improving the soil biology, increasing the diversity, and moving that from a bacterial dominant to fungal dominant system. You can see 777 pounds of nitrogen per acre was captured out of the atmosphere just with the biology. This is how we're being able to look at a soil now. You can see this is analysis of that particular experiment of the control and the compost. We're able to look now at the nitrogen fixation, both symbiotic and free living. We're able to look at nitrogen cycling. We're able to look at carbon cycling in this. And carbon cycling is really important because this is a living system. We need to realize that a certain amount of this is going to have to go into metabolism. A certain amount is going to be going into other organisms in the soil. We also look at metal oxidations. There's organisms in here that will mine any nutrient that plant needs. There's even some in here that can oxidize gold, the most stable element on this planet. We'll also see phosphorus solubilization, antibiotic and antimicrobial compound secretors. We see see quorum sensing, and we also see CO2, carbon monoxide oxidation, degrading pesticides and xenobiotics, and also phytochemical production. If you're curious, this chart back here makes up about 195 species that have set the treated soil against the control. You can see all the different aspects of how microbes make this plant system work. We also looked at the metatranscriptome to see what proteins were being transcribed at the time. As you can see, quorum sensing was one of the highest. It was like 20 times higher than any other protein being transcribed in this process. Quorum sensing is when you get a community of organisms that get together, start to express genes that they don't express or can't express as a single organism. It's like when we, as a community, get together to make a change that one person can't make, but a whole community working together can. What we've seen also in this is an increase in soil carbon use efficiency. Most scientists are going to tell you that if you increase soil carbon, you're going to increase respiration. It's true to a point, as you can see, but it starts to level off. When you look at this, there's an 18 times increase in beginning soil carbon mass. There's a 5 times increase in soil and microbial biomass in this system, but only a 4 times increase in the carbon respiration mass. This is how nature does this. This is how nature is able to put carbon back into the soil. As she improves, going from a bacterial dominant, low carbon soil to a high fertility, high fungal to bacterial ratio, high carbon soil, the dynamics on how carbon respiration in the soil changes. If you look at this a different way, this is the same experiment, 18 times increase in soil carbon mass, and only a 4 times decrease in relative soil carbon respiration. If you look at the total carbon respired from, total original carbon respired from this, in that bacterial dominant soil, 55% of that carbon is respired. But in a fungal dominant soil, you can see only 11%. That's your 4 times decrease. It increases carbon captured in the soil if you have this biodiversity, the biological diversity. Over the first 4.5 years of this experiment, we were able to capture carbon at the rate of 0.25% increase in the top foot per year. That's 10.7 metric tons of carbon per hectare per year, or 37 metric tons of CO2 per hectare per year, pulled out of the air and put into the soil. Other scientists looking at, like West, looking at 67 long-term no-till studies, and Nigley with agroecosystems using mulching, and Dr. Law with arable cropping systems, they see only half a ton to 0.7 tons carbon per hectare per year. We've seen with the right biology and the diversity of crops put on there, a 0.25% increase per year with the potential of 0.5% a year. Now, Gabe has already shown us that he can do over 0.5% per year. At 10.7 tons carbon per hectare per year increase, it only takes 40% of the arable land on this planet to capture all anthropogenic CO2. So at 0.5% per year, we're looking at half of that rate. In the process, we're also restoring the micronutrient availability of these soils. What we've done in our agricultural systems by adding all these synthetic fertilizers, we've outstripped the ability of these soils and the microbes in the soil to furnish the micronutrients. This is a 20-month study, the 5 sampling periods on when we first started applying beam on these soils. We saw 1,100% increase in manganese, iron, 83% increase in magnesium, 76% in calcium, 62% in zinc, and 40% in copper. We also saw nitrogen, phosphorus, and potassium as well increase in this. Again, we're just growing plants and inoculating with the right biology. And we've only inoculated once on this 7-year trial. So we're also improving crop yields in the process. A preliminary study on cotton production was a 114% increase, or almost a doubling of the availability of growing cotton. In our chili production, we do chilies in New Mexico, saw a 98% increase. This is this year's cotton, on July 17th, August 2nd, August 29th, a little over 6 feet high. Now, just because you have 6 feet cotton doesn't mean you have a lot of cotton. We did 5 bales of cotton on this plot, 2,200 pounds of lint, and 3,300 pounds of seed per acre. That's about a $2,000 yield, gross, on a crop that you use no fertilizers, no pesticides, and no herbicides. This is just biology that makes this grow. So we can meet the challenges ahead of us. We can produce more food. We can reclaim degraded soils and increase land area. We can use less water by increasing both plant water use efficiency and soil water storage capacity. We can reduce energy use by allowing nature to extract and fix the elements. We could also reduce both agrochemical pollution and atmospheric CO2 concentrations at the same time. Why should we pursue this? There is no planet B, and this is as good as it gets. So align yourself with nature.